guys, I hope you all are doing well at home and enjoying our online lecture videos also. So I request you to subscribe our channel NUCLEIN Biotech by pressing the bell icon given below for more such videos. And also don't forget to like, comment and share. Now our today's topic is amino acid. In the last lecture regarding amino acid, we talked about the tricks how to learn different amino acid according to their classification, and today again we discuss more about amino acid and their properties. Now, what is amino acid? Amino acids are organic molecules that when linked together with other amino acids form a protein. Amino acids are essential to life because the proteins they form are involved in virtually all cell functions. Some proteins function as enzymes, some as while others provide structural support. Although there are hundreds of amino acids found in nature, proteins are constructed from a set of amino acids. These are 20 in number. Now, in terms of biochemistry, amino acid is defined as amino acids are a group of organic compounds containing two functional groups, amino group that is NH2 which is basic in nature and carboxyl group which COOH which is acidic in nature. Now let's talk about the general structure of amino acid. Amino acid have the following structural properties: a carbon that is alpha carbon, a hydrogen atom, a carboxyl group COOH, an amino group NH2 and a variable group or R group. All amino acids have the alpha carbon bonded to a hydrogen atom, carboxyl group and amino group. The R group varies among amino acids and determines the difference between these protein monomers. The amino acid sequence of a protein is determined by the information found in the cellular genetic code. The genetic code is the sequence of nucleotide bases in nucleic acid, that is DNA or RNA, that code for amino acid. These gene codes not only determine the order of amino acid in a protein, but they also determine a protein structure and function. Now next is the optical isomer of amino acid. All amino acid except glycine, which has two hydrogen atom on the alpha carbon, have an asymmetric alpha carbon, as may be seen in the figure given below. One can therefore write for example, in the case of alanine, the septal formula of two optical isomers non-superimposable because one is a plane mirror image of the other and whose physical and chemical properties are identical. One therefore distinguish for each amino acid an isomer D and an isomer L depending on whether the septal structure is identical to that of D glyceraldehyde or L glyceraldehyde, whose absolute configuration is known. The septal formula being rather complicated, the plane projection formula proposed by Fischer is preferred. By convention, one writes the amino acid of the D series with the amino group on the right and those of the L series with the NH2 on the left. It may be noted that the prefixes D and L have absolutely no significance related to the direction in which the plane of polarized light is rotated by the amino acid. The direction may be specified by a sign plus or minus placed before the name of amino acid. For example, L for minus sign for leucine, keeping however in mind the fact that the rotatory power changes the condition of the medium, particularly the pH. All amino acid constituting the proteins belong to the L series but in various bacterial peptides, particularly those present in the cell wall or certain peptides having an antibiotic activity, one finds amino acid of the D series. On the other hand, certain amino acids have an additional asymmetric carbon in the side chain, which increases the possibilities of isomerism. Thus, for threonine, one will have D and L threonine and D and L allothreonine. Let us note that in organic chemistry, the designation L and D are now replaced by the letter S and R. For example, S-alanine and R-alanine. Now next is the classification of amino acid. So these are based on structure, polarity, nutrition requirement and metabolic fate. Now let's discuss them one by one. First is based on structure, amino acid with aliphatic side chain. So some of the examples are glycine, alanine, valine, leucine and isoleucine. Next is the amino acid containing hydroxyl group. Examples are serine and threonine. Next is the sulfur containing amino acid. Examples are cysteine and methionine. Next is the acidic amino acid. Examples are aspartic acid, asparagine, glutamic acid and glutamine. Next is the basic amino acid. Examples are lysine, arginine, histidine. Next is aromatic amino acid. Examples are thiosin and tryptophan. Next is amino acid. Example is proline. Now classification of amino acid based on polarity. So polarity reflects the functional role of amino acid in protein structure. So they are further divided into two groups: non-polar amino acid and polar amino acid. Non-polar amino acid referred as a hydrophobic, no charge on R group. So some of the examples are alanine, leucine, isoleucine, valine, etc. Now polar amino acid with no charge on R group, they possess groups such as hydroxyl, amide and participated in hydrogen bonding of protein structure. Some of the examples are glycine, serine, threonine, cysteine, etc. Now polar amino acid is further divided into two: polar amino acid with positive R group. Some of the examples are arginine, lysine and histidine. Again, polar amino acid with negative R group. Examples are aspartic and glutamic acid. Now classification based on nutritional requirement. This is further divided into two groups: essential and non-essential amino acid. So amino acid which can't be synthesized by body are known as essential amino acid. These are need to be supplied through the diet. So and it is required for proper growth and maintenance of the individual. So some of the examples are arginine, valine, histidine, isoleucine, leucine, lysine, etc. Now next is the non-essential amino acid. So these amino acids can be synthesized by the body, so meet the biological need and no need to consume in the diet. So some of the examples are glycine, alanine, serine, cysteine, etc. Again, amino acid classification based on metabolic fate. Now carbon skeleton of amino acid can serve as a precursor for the synthesis of glucose, that is glycogenic or fat, ketogenic or both. So these are further divided into three. So first is glycogenic amino acid that can serve as precursor for the formation of glucose or glucogen. Some of the examples are alanine, aspartate, glycine, methionine, etc. Next is ketogenic amino acid. Fat can be synthesized by this amino acid. Examples are leucine and lysine. Next is glycogenic and ketogenic amino acid. So in this isoleucine, phenylalanine, tryptophan, triazine are the precursor for synthesis of glucose as well as for the fat. Now 21st amino acid is selenocysteine. It is found at the active site of certain enzymes or protein, selenoprotein. Incorporation of selenocysteine into the protein during translation is carried out by the codon, namely UGA. UGA is a stop codon that terminates protein synthesis. It generated from serine directly on the tRNA and thus incorporate into proteins. Now 22nd amino acid is pyrrolysin. It is stop codon. UGA can code for pyrrolysin. Now take a look to the properties of amino acid. First is the physical properties. Now solubility. Amino acids are soluble in polar solvent such as water and ethanol, but they are insoluble in non-polar solvents such as benzene or ether. If we talk about the melting point, high temperature is required to melt, which is 200 degrees Celsius. Now uh, if we talk about taste, glycine, alanine, valine are sweet in taste, leucine is tasteless, or arginine is bitter. Monosodium glutamate is a flavoring acid. Now move on to the optical properties. Except glycine, all amino acid possess optical isomers due to presence of asymmetric carbon. Some amino acid have a second asymmetric carbon, for example isoleucine and threonine. Now amino acid as ampholytes. Amino acid have both acidic as well as basic group. They can denote a proton or accept a proton. So these are known as ampholytes. Now Zeuter ion or dipolar. The ion at isoelectric point which carries positive and negative charge is called Zeuter ion. In acid solution, Zeuter ion combines with H positive ion to form base yielding cation. When the current is passed, the amino acid migrates towards cathode. In alkali solution, Zeuter ion combine with OH negative to form acid yielding anion and migrates towards anode during the charge of current. Zeuter ion is the ampholyte, that is, it is both a proton donor and proton acceptor. The acidic and basic strength of the Zeuter ion are different, and a solution of pure amino acid in water is not neutral. Proteins like the amino acid contain 6 acidic and basic group exist in solution as Zeuter ion. Now next is isoelectric point. The isoelectric point, or we can say isoelectric pH of an amino acid, is that pH at which it has no net charge and does not move in an electric field. So for example, isoelectric pH of aspartic acid is 2.9, lysine 9.7 and arginine 10.8. It has a significant value in the clinical laboratory to determine the mobility of known compounds in electric field. It selects appropriate buffer for separation of one from another. Proteins like amino acid have isoelectric pH at which they are least soluble and migrate least in an electric field. Above pi, or we can say above isoelectric pH, protein act as acids and from negative protein ion, whereas below isoelectric pH they act as bases and form positive protein ion. Now move on to the titration of amino acid. Now first is titration of a neutral amino acid. When acid HCl is added, the base R COO negative is titrated Now next is the titration of amino acid. When acid is added, the base is titrated, and when a base is added, the weak acid is titrated. So the existence of different ionic forms of amino acid can be understood by the titration curve. Now, this is a graphical representation of leucine. At low pH, leucine exists in fully proteinated form as cation. As the titration proceeds with NaOH, leucine loses its protons, and at isoelectric pH, it becomes a deuter ion. Further titration results in the formation of ionic form of leucine. Now after physical properties, we're gonna discuss about chemical properties. Journal reaction of amino acid are mostly due to presence of two functional groups COOH and NH2. Now first is reaction due to COOH group. In this first is formation of salt. Amino acid form salt with base and ester with alcohol. Next is decarboxylation. It is carried out in the laboratory, but it takes place by enzymes action in living cells. And decarboxylation is used to produce amines. So some of the examples are histamine and gamma amino butyric acid, that is known as GABA, GABA, from amino acid histidine and glutamate. Now reaction with amino carboxyl group of dicarboxylic amino acid react with NH3 to form amide. For example, aspartic acid is when reacted with NH3, it forms asparagine. Same glutamic acid is reacted with NH3, it forms glutamine. Now second is reaction due to NH2 group. Amino acid behave as bases and combine with acid to form salts. Now next is reaction with ninhydrin. Alpha amino acid react with ninhydrin to form purple, blue or pink color complex. So for example, amino acid when combines with or reacts with ninhydrin, it produces keto acid plus amino plus carbon dioxide plus hydrodentin. And when this hydrodentin reacts with NH3 and ninhydrin, it forms rumen purple. It is used for quantitative determination of amino acid and protein. Like protein and hydroxyproline gives yellow color with ninhydrin. Now next is color reaction of amino acid. Amino acid can be determined by specific color reaction. Next is transamination. Transfer of amino acid amino group to form an amino acid to a keto acid to form new amino acid. It is a very important in amino acid metabolism. Next is oxidative deamination, and it liberates free amino. At last, if you have any query regarding this topic, you guys can WhatsApp on the number given on screen. Thank you.